So I've gotten several emails from people thanking me for the change in policy. So if you haven't seen the change in policy, I have changed the policy for cell phones, which might be a good time to check and make sure you've got it in vibrate mode. I decided that rather than penalize everybody because a few people are not being responsible with their phones, that I would penalize the irresponsible people. So the policy on cell phones goes as follows. If you have a cell phone go off with a ringtone, and I hear it, then I will confiscate your phone and you will lose 10 points off of the next exam. So, good incentive to turn the ringtones off. It might be a good idea to just leave them off all the time. That way you wouldn't have to worry about that. But that's just my personal preference. Okay. So, please be aware of that policy. Please turn off your ringtones. All right, so I'm a little bit behind, so I want to get into the material and talk about more about protein characterization. Okay? So the first thing I want to talk about are some techniques that we use to characterize proteins. And the first of these that I'll talk about are immunological ones. This first one is a very popular and very commonly used technique in laboratories. So imagine that you've run a protein gel, and you've got this whole series of bands. And remember, if you're purifying a protein, you have to have some way of knowing which one's yours. One of the ways of knowing which one's yours is if you have an antibody that will react with it. That is, not react with it, but combine with it. Okay? And that's what the basis of this technique is. So I'm going to step you through it. There's a lot of iterations on how this actually is done. So if you've done it in a lab and it's a little bit different, don't be too surprised. But the basics are the same. It relies first upon the fact that antibodies, which are proteins in our immune system, recognize and bind to very specific structures. Antibodies recognize and bind to very specific structures. Okay? Now, these very specific structures, of course, are at the molecular level. And so in your immune system, if you have, let's say, a virus that infects you and it has a characteristic protein structure on its coat, then your immune system will make... That's interesting. Lick me. Okay, well... This would suggest to me that lick me is somewhere in here. Could lick me perhaps turn off your Bluetooth? Okay. I like that. That could be a comment on my cell phone policy too. You never know. So, all right. So the point of this is that you have antibodies in your immune system, and they're there because they give you defense against invading organisms and invading viruses. These invading systems have specific structures in their proteins, and your immune system recognizes those, binds to them, and takes them out of action. So when we're doing these techniques, we're actually purifying these antibodies that recognize specific structures. And typically, we purify them from a bunny rabbit. It's not really harmful to the bunny rabbit. We have to take a little bit of blood from the bunny rabbit, but we don't kill it or anything in the process. It's a fairly innocuous process for the bunny rabbit. And the bunny rabbit makes us lots of nice antibodies. So let's say I've got a protein. I want to follow this protein perhaps through my purification system. Perhaps I've already purified it, and I want to see where it shows up on a gel. I take my protein, I inject it into a bunny rabbit, and the bunny rabbit says, whoa, this protein, I haven't seen this before, so I start making antibodies against it. The antibodies are produced. I now grab that blood from the bunny rabbit, and I purify the antibodies out of it. So I've now got antibodies that will recognize and bind to the specific structures in my protein of interest. That's the first step. Okay? Now, I'm going to simplify this process for you somewhat. And what I'm going to do is tell you a sort of a shortcut way of doing this method. It's not commonly done, but it's a shortcut way of understanding. We don't need to know all the steps of the method in order to understand the basics about what the method is teaching us. First of all, the thing that an antibody binds to is called an antigen. So the protein that I am interested in, that I injected into the antibody, is an antigen. The antigen is, of course, the structure that's recognized by the antibody. Well, I want to use this antibody to tell me on a gel which protein's mine. There's thousands of bands on this gel. Which protein is mine? I would like to see this. I might like to know, okay, this protein's mine, but is this protein that's mine being made in this cell type that might be liver, and also in this cell type that is, for example, skin or brain? So I could run extracts of each of those and probe it with the same antibody and ask the question, is this protein present in all 3 or 4, 10 of these extracts? So it's a very powerful technique for telling me really the presence or absence of my protein of interest in an extract. Well, to do this, I have to have some way of visualizing things. Antibodies by themselves don't visualize. Okay? They're fairly invisible. You look at them, there's no color, there's no any other thing associated with them. So there's a variety of methods of visualizing those. For our purposes, I'm going to say that we're going to take that antibody and we're going to put a little fluorescent marker on it. Okay? A little fluorescent marker, meaning a red dot, a green dot, or something like that, such that when the antibody is present, I will see a little band that's the color of that fluorescent marker. And when the antibody is not present, of course, I won't see that color. So the only thing that brings the color to the process is the antibody itself. So here's just a labeling, and you can see in one case where they have used this technique to study an embryo. And they've got a flagged antibody, and they took this embryo, this developing embryo, and they probed it with this antibody for a specific protein being made during development, and look at the segmentation that they see. Okay? So we see in an embryo, for example, this is probably a fruit fly if I recall correctly, we see in an embryo that what happens is that there's a specific pattern of location in an embryo where proteins are being made and not made. We can see there's a lot more of the protein being made right here, not so much of the protein being made here. Now this is not a Western blot. This is simply using the antibody to look at an embryo. Got an embryo sitting here, add antibody to it, wash away the unbound stuff, and this is what I would see in the microscope. If I want to use this antibody for Western blotting, then I have to do a couple of additional things. So here's what I do. First, I take my mixture of proteins that I'm interested in. So let's say I've got a mixture of proteins from liver, from skin, and from brain. Okay? And these proteins I separate on different lanes of a gel. One lane might correspond to liver, another lane corresponding to skin, another lane corresponding to brain. I do SDS-PAGE just like I've been talking about before. So I separate the proteins on the basis of size. And I then take this gel and I do what's called a blot. And in this blot, what I'm doing is I'm transferring from the gel onto a piece of paper those proteins. This is typically done with an electrical current. So I can actually put a plate on top of there that has an electrical charge and drive the proteins out of the gel and onto the paper. This paper is designed so that the proteins will stick to it. So now on this paper, I have an exact representation of what was on that gel. So that's the blot part of it. I've just blotted the proteins onto the paper. I then take that paper and I do what's called probing, which simply means that I add my antibody in an appropriate mix where it will recognize specific structures. And even though that protein's been denatured, amazingly enough, it usually will still be bound by the antibody. Okay? The antibody will recognize my protein of interest, but it's not going to recognize other proteins because they don't have that specific structure. And as a result of that analysis, if everything goes well, I see at the location on the gel of my protein, I see that band corresponding to that fluorescent dye that I had. In this case, I had a red one, let's say. Actually, I guess it's sort of a gray one there, but you get the idea. So now I can ask the question, is this protein present in my extracts for the skin? Is it present in extracts for the brain? Is it present in extracts for the liver, et cetera, and answer that question very quickly. Now, this is an additional tool to the 2D gel. In the 2D gel, we said we saw all these proteins at once. Okay? 2D gels are very powerful, but they're also very, very technically difficult to do. This is a much simpler process, and it's specific for my one protein of interest. So this is used in addition to 2D gel electrophoresis to flag proteins of interest to us. Okay? Now, when I run a system like this, I need to have some appropriate controls, right? So what are the controls I would have on a Western blotting gel? What would I need to know to tell me my experiment is working? I can think of at least two things. So what's a standard compared to? What is that? What's that? Come on, you guys are scientists. So I have the known. So I take my known protein that I know how to bind that antibody, and I put one lane of the gel so that it's got that. If I don't put that on there, and I don't see anything on the gel, what can I conclude? Nothing. I can't, you'd say, well, there's no protein there. Well, how do I know I didn't screw up and it wouldn't bind my protein even if I wanted it to? So I need to have a known protein on there that it should bind on that gel. So that's what's called a positive control. I need to have a negative control as well. What's a negative control? What would I use as a negative control? So a protein that wouldn't bind to it, and or maybe an extract that wouldn't bind. Let's say I'm studying human liver, and I decide, well, I've got a bunch of bat liver laying around. Let's take this bat liver and put it in there and see, right? So presumably there's enough difference between that bat liver and human liver that I wouldn't see it. What if I saw a band in the bat liver and I saw it in the human liver? What's that? It could be the same protein. It could be closely enough related that I'm seeing the same structure. I'd have to do additional work if I saw that happen. So in the ideal experiment, my negative control would show nothing. My positive control would show the band that I thought should be there. And then I could interpret what I saw elsewhere. If I saw no bands, I'd say, wow, my protein isn't present in detectable amounts in these extracts. Or maybe it's present in brain, but not present in liver. That's kind of interesting as well. Make sense? Everybody understand that? Okay. So that's how a Western blot works. Very powerful technique. There's different ways of visualizing that. So I talked about putting a color marker on there. Sometimes they use light reagents such that light is produced and a variety of different things that happen. So we don't need to worry too much about the details of that. Okay, the next technique I want to talk to you about is an incredibly powerful technique called Maldi Tov. Okay, Maldi Tov. So Maldi Tov is an acronym for a mouthful of mumbo jumbo involving lasers and so forth. The bottom line about Maldi Tov is that it's a technique that allows us to determine the mass of things very quickly and accurately. So it's a mass spectrometric technique. A mass spectrometer is something that determines masses, and Maldi Tov allows us to determine masses of peptides. Now, I told you before that a lot of the techniques I talk about don't work very well on large proteins. This is a real good example. This is one of those times when we want to take that protein and break it into pieces because Maldi Tov doesn't work real well, at least not real accurately, on giant proteins. We want to break it up into smaller, easily analyzed proteins. Okay? So we're going to use proteases. We're going to use cyanogen bromide to break that guy into smaller pieces so we can do it. Well, how does this technique work? Well, a mass spectrometer, the TOF on the Maldi Tov stands for time of flight. Okay? Now, time of flight is really critical to understanding, or really critical to the determination of mass in this technique. I've got a protein. I've got a protein that I've digested into pieces, and I've got one of those pieces sitting and I'm ready to analyze. I want to know what the mass of that piece is. And we'll see why that's important in a minute. But I want to know what the mass of that piece is. So I have a system set up where that piece is placed in this vacuum chamber. This is a vacuum chamber. And the piece is sitting on a little tiny, what's called a probe. Okay, it's just a little, you want to think about it a little, almost like a pin, a little thing that's sitting in there. My protein or my peptides of interest have been dissolved in a liquid, and then the liquid has been evaporated. So there's a little crystal of this protein sitting there on this probe. I put it into the mass spectrometer, and at first nothing happens. It just sits there. Okay? There's nothing to force it off. But then I take a laser, and that's the L part of Maldi Tov. And no, you don't need to know all these things, okay? At least all those names. You need to know the process. A laser is shined at that probe. Now what that does is it causes that crystalline material on there to volatilize, meaning it goes into a gaseous phase. And in the process of doing that, the peptides that I have in there ionize. Okay? The peptides ionize. In this case, we show them as gaining a charge, a positive charge. Now, that positive charge becomes the basis of our analysis. We know the exact instant that we hit it with the laser. We have a positive charge, we have a vacuum chamber, so there's no air in the way of these ions moving in the chamber. We put a little positive charge down at this end. We have a negative charge down here that has a detector that simply says, okay, something just hit me, something just hit me, something just hit me. Right? Now all we have to do is time how long it goes from the time that we shine the laser on there until the time that the detector says something hit me. Let me ask you first, if they all have a +1 charge, and I have different sizes of peptides in there, which one's going to travel the fastest? The smallest, because the charge to mass ratio is greatest when the size is smallest. I can calibrate my instrument, and I can determine in that instrument the size of each one by how long the time of flight is for that ion to get there. It's a very cool technique. You might say, well, why, if you've already got it purified, why do you have a mixture of these? The reason you have a mixture is because these ionic compounds tend to break down. They break into pieces, and they break into nice, convenient pieces about at every peptide bond. So now I get a series of pieces that differ from each other by one amino acid. If I'm really careful, and I say, okay, here's the first one that came through, it had a mass of 5,112, and the next one that comes through has a mass of 5,217, I can subtract those two masses and say, oh, that corresponds to a tryptophan. The difference between these two was a tryptophan. So I'm very careful. I can actually do this entire analysis, and I can determine the sequence of the peptide that was in there. That's incredibly powerful. So Maldi Tov allows me to determine the sequence of a polypeptide chain. Wow. Well, if I know the sequence and I have the genomic sequence of the organism, I can ask the computer, well, where in the genome do we find tryptophan, followed by cysteine, followed by alanine, followed by glycine, followed by glutamic acid? And the computer says, oh, well, there's only about 3 places in the genome it appears. One is a DNA polymerase over here. Another one is a kinase over here. Another is a phosphatase over here. Then I can do additional experiments and very quickly determine what that protein was that was on there. Now, the beauty of this is that this process is actually automated. It's automated. So, as an example, I could take and I could feed into a robotic system a 2D gel. And the robot would do a nice little job of cutting out each little spot, digesting it, running the analysis, and the computer would do the work while I'm going to get coffee. Very good thing, if I drink tea, but other than that, it's a good scheme. Okay? Now, while that's going on, okay, I'm learning the location of every protein in the genome on that 2D gel. That's how I know where my proteins are. Okay? A robotic system can determine in a single day 6,000 proteins on a 2D gel. When I first learned that, I thought, automation is amazing. Absolutely amazing. Back in the dark ages, when I was working on my master's degree, I did a lot of mass spectral analysis. And if we got a single mass spec of a compound that was very simple in size, that was considered a pretty good month. Okay? That was a pretty good month. Yeah, question? Yep. So all I know is that each one differs in size by 1 amino acid. So all I have to do is just look at that sequence that comes and hits it. So if the first one coming through is a glycine, and the next one coming through is a glycine plus a tryptophan, and the next one coming through is a glycine plus a tryptophan plus an alanine, you see, I know what the sequence of that thing actually is. Does that make sense? No. All right, so let's say, so I do my analysis. Something comes through and it's got a mass of, I don't know, 71. I'm just making this up because I don't know what the masses of the amino acids are. My computer says glycine. That's followed by one that has a mass of 171. And my computer says the difference between 171 and 71 is 100, and the only thing that has a mass of 100 is cysteine. So now that meant that second one had to be glycine and cysteine.
What if two glycines come out?
Then that would mean I'd have glycine followed by glycine.
What if the original polypeptide had a glycine at the very end? When at that very end glycine come out?
So they're breaking sequentially. So they're breaking sequentially. That's the key here. Okay. So if you do this very carefully, you break it sequentially, you see the sequence as they hit the detector.
Okay, so it breaks the line. It doesn't like break it all at once.
That's right.
That can happen too. So I'm giving you the simple case. It turns out that it does break into pieces, and it takes a very sophisticated computer analysis to pull that out, but it can. But I'm just giving you a simple case so that you can understand how sequence could be read using this. Does that make sense, though? Yeah. So I just simply look at the difference, and wow, all of a sudden I've got the sequence of my polypeptide. It's really awesome. Okay, so that's Maldi Tov, and Maldi Tov is an incredibly powerful technique. The last things I want to say about characterization of proteins relate to two techniques, and there are entire courses that are taught about these, so I'm not going to do much more than just say simple things. The first is known as X-ray crystallography. X-ray crystallography really is an incredibly powerful technique, and it's probably the most important technique for us to determine protein structure. The way it works is you have to have a pure protein. So you have to go through all the different techniques, all the processes of purifying your protein so you get the one that you are after. And it has to be absolutely pure. It can't have other junk in there. If it's got other junk in there, it's going to screw up your analysis. You then take this protein, which is now in solution, and you allow that solution to start drying so that the protein becomes more and more concentrated. As it becomes more and more concentrated, eventually it will precipitate out, and it will make a crystal in the ideal world. Now, crystals can form in many ways, but if we get a good crystal, we can actually do analysis on it. And making a good crystal is more voodoo than it is science. Okay? It really is. Once you get a given way of making that crystal, you're set. But sometimes finding the ways to do that, it's not really understood what needs to happen to make a good crystal for X-ray crystallography. Suffice it to say, I've got my good crystal. This crystal now I take and I place into an X-ray crystallographic spectrometer. Blah, blah, blah. It's sitting on a little probe, just like it was in the mass spectrometer. Only now it's a crystal that's sitting there, and it's a pure crystal. That crystal is perfect. Everything is oriented in a very, very regular fashion. Okay? That's one of the things I have to have for a crystal to be good in an X-ray crystallography machine. If I have a good one, I can shoot X-rays at it from a variety of angles, and it turns out X-rays get diffracted, that is bent, by interaction with electron clouds. The more dense the electron cloud, the more diffraction. So I have a machine that simply measures the pattern of X-rays that comes out from my crystal, and then we get basically an electron density map of where all the densities were inside of there. Okay? Who's got it? Where's it at? So, the computer takes this pattern of X-rays that come out. Okay? And the pattern of X-rays that come out were based on the electron densities of the substance that was there. Okay? That pattern of X-rays, ultimately, I have to take an electron density map and say, where were the individual amino acids contained inside of there? So I might say, okay, this really looks like I've got a glycine right here. This must be a glycine. That thing next to it, I'm not really sure. Could it be alanine? Because alanine sort of looks like that if I twist it this way. Or perhaps it could be serine if I twist it that way. Now we start seeing where the Ramachandran plot becomes useful because I know what twists are allowed. And I can start building my protein structure using knowledge about how these things can possibly twist. Okay? So these are very, very useful things for me to know as I'm building a structure. Okay? So X-ray crystallography ultimately will tell me the position in three-dimensional space of every atom in a protein that has thousands and thousands of atoms. That's an incredibly powerful technique. Incredibly powerful. Okay. Let's not talk about NMR. NMR is, I'll just briefly say about NMR, another analytical technique, another spectroscopic technique uses magnetic resonance. Yes?
Quick question.
Uh-huh.
With crystallography, is that basically just to find out what the sort of building configuration of what protein is?
So with crystallography, you literally build the configuration of proteins that are consistent with the spectra that you see. That's what crystallography ultimately is about. Yes?
How can you see each individual molecule when they're all packed on top of each other?
What you see are densities, and it's a very good question. You actually get three-dimensional information by looking at the different angles of this. So you can look at it from above, below, the side, and so forth. And ultimately, you can see that. But as you can imagine, it's an enormous problem. So that having help in terms of what might be here, 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 here is a very big factor in the process. Okay, so nuclear magnetic resonance uses magnetic resonance. This arises from the fact that nuclei have spin. We don't have to worry too much about that. The advantage of nuclear magnetic resonance over X-ray crystallography is we can do the analysis in an aqueous solution. We do not have to have a crystal. And making a crystal can be a limitation for a lot of different X-ray crystallographic techniques. Okay? So that's a very, very powerful thing for us to have. Okay, enough said about that. Questions about that? Okay, so that finishes what I want to say about protein characterization. Let's go talk about our next topic, which I think most people interested in health sciences are very interested in, and that's oxygen-binding proteins, myoglobin and hemoglobin. Now, we all know that hemoglobin carries oxygen in our blood. Okay? What you probably don't know, you probably haven't heard much about myoglobin. What is myoglobin? Well, myoglobin's got the same globin name, and it turns out it has a lot of similarity to hemoglobin, but not completely. So myoglobin is also an oxygen-binding protein, just like hemoglobin is an oxygen-binding protein. Myoglobin's characteristics, however, are different from hemoglobin's. Hemoglobin has 4 subunits, 2 chains called alpha and 2 chains called beta. 2 identical alphas, 2 identical betas. It has quaternary structure. Myoglobin, on the other hand, has only a single chain, so it doesn't have quaternary structure. Both the single chain of myoglobin and the alpha and the beta chains of hemoglobin contain a group called heme. Now, heme is an iron-containing group that actually does the oxygen binding. So hemoglobin has hemoglobin has a heme group just like myoglobin has a heme group. Okay, now what's the difference then? Well, the difference, as we're going to see, is that myoglobin is really good at binding and storing oxygen. Okay? And hemoglobin is really good at binding and letting go of oxygen. Now, myoglobin will also give up oxygen, the difference being that it takes a very low oxygen concentration before it will do so. I like to think of myoglobin as kind of like a UPS, okay? An uninterruptible power supply. You have these in your computer, right? You plug it into your computer, and when the power goes off, it keeps the power going so you can shut your computer down. The UPS of myoglobin, what it will do is it will, in fact, losing my mind here. Nothing new there. When the oxygen gets very, very low, then myoglobin lets go of its oxygen, but only when it gets very, very low. So it's kind of like an oxygen battery. It's giving it up when things get very, very low. We don't want it to get so low for hemoglobin. We want hemoglobin to give up its oxygen fairly readily. We don't want our cells starving to death, basically. Okay. So I'm throwing a lot of things at you. Here is what a heme group looks like. Okay? You should be able to draw that on the first exam. No. Okay. So heme has at its very center an iron atom. That iron atom is in the +2 state. Very important component. If iron gets oxidized to the +3 state, it will not bind oxygen. So first of all, we have to have iron in the +2 state. Second, this guy is embedded inside the protein. So myoglobin has one of these, hemoglobin has one of these, etc. Okay. Now, let's take a look at that from 3 dimensions. Okay. Now, I think you're going to find, as most students do, and certainly as I do, that this heme group is one of the most interesting groups in all of biology. You just took hemoglobin for granted, probably in your life. Okay. So most of what I will have to say has to do with hemoglobin, but this very first thing has to do with both. If we were to look at the heme group inside of myoglobin and hemoglobin, what we would see is the following. First of all, that flat plane that we saw back here, if we're looking at it top down, and now we twist it to the side, we're going to see the next view that I have for you here. And when we twist it to that side, what we see is it looks like this. So we're looking at it edge on, there's that iron atom, and we see that the iron is attached from below to a histidine. Now, this is interesting. Look at this and compare it to this. Over on the right, we have that same group, and that same group has been bound by oxygen. In other words, it's grabbed a hold of an oxygen. Now, there's a very subtle change that has happened in this binding of oxygen. A very, very subtle change. That iron has been moved up by 0.4 angstroms, a very, very, very tiny amount. The effect of moving that guy by 0.4 angstroms has an enormous effect on hemoglobin and none whatsoever on myoglobin. Okay? So everything I'm going to be talking about with respect to hemoglobin arises ultimately from this little tiny movement here. So oxygen literally pulls it up. Okay? Now, well, how can such a tiny change have such a big effect? We're going to see that it really has a big effect in hemoglobin. And I want to do the old thing about the toe bone being connected to the foot bone, connected to the ankle bone, connected to the shin bone, connected to the knee bone, connected to the, right? If you pull on the toe, what happens to the leg? You see changes happen, right? You can't pull on that toe without having some effect going on. And in fact, there's the toe right there. That iron is the toe, and it's getting pulled up slightly. This histidine is attached to the rest of the globin, whether it's myoglobin or hemoglobin. So globin is the protein that it's attached to. This amino acid gets moved slightly. The amino acid that's attached to it gets moved slightly. Attached to it gets moved slightly, etc. So pulling on the toe of this guy really has the effect of changing everything else about this. And the changes are very, very tiny in physical space, but enormous in terms of property. Okay. Deoxyhemoglobin, of course, has no oxygen. Oxyhemoglobin has oxygen. Notice that it's O2 that's being bound. Okay? And we'll not talk about that. Okay. And I'm not going to talk about that at the moment. Okay. If I look at the binding of oxygen by myoglobin, what I discover is that it looks something like this. What this is fraction of saturation. So if I get up to 1, it's 100% saturated. And over here, down here along the y-axis, is the oxygen concentration. This guy is really good at binding oxygen because it takes very low concentrations of oxygen to get 75 to 80% saturated. When I say 75 to 80% saturated, I mean 75 to 80% of the molecules have an oxygen. Each one binds 1. So 75 to 80% of the molecules have an oxygen at very low concentrations. Myoglobin is very good at holding on to oxygen, but it's not going to give it up until the oxygen concentration starts to fall very, very low. And when it starts to fall very, very low, then myoglobin gives up that oxygen and the cells have oxygen. This is an oxygen battery. A UPS is something that stores electricity until the power goes out and you need it. Myoglobin stores oxygen until the cells start starving for oxygen and you need it. Okay? Everybody with me?
This graph shows myoglobin, not hemoglobin.
This is just myoglobin. That's right. Hemoglobin is significantly different. Here's what hemoglobin's curve looks like. Now, hemoglobin, at the same concentration of oxygen, way down here, hemoglobin has virtually no binding. Okay? That's what percentage of those molecules have oxygen, very tiny percentage. Myoglobin has 80%, where hemoglobin has essentially none. However, as the oxygen concentration starts increasing, more and more hemoglobin starts getting oxygen until we get up to the high concentrations that we would find approximately in the lungs, and guess what? They have essentially the same amounts. Now, why is this important? This is important because hemoglobin is much more willing and ready to give up oxygen than myoglobin is. Myoglobin is not giving up oxygen until it really runs dry of oxygen. Okay? Hemoglobin, all right, is giving it up sooner. All right? At the same concentration of oxygen, look how much hemoglobin has given up. Myoglobin is still clinging to it. Okay. So hemoglobin is good at picking it up and delivering it. Myoglobin is good at storing it and giving it up only when it has to. Okay? The big kid and the little kid with candy, right? The big kid takes the little kid's candy, hangs on to it, right? Until somebody puts, you know, you're going to make you stay after school, then the big kid's got to give it up. Okay. All right. Well, what we're seeing with hemoglobin is we're seeing that its percentage of oxygen bound as a function of oxygen concentration varies. And it varies significantly. And it turns out that when we look at this, we discover that there's different binding states. These different binding states we refer to as cooperativity. Cooperativity. It turns out that at very low concentrations of oxygen, virtually no hemoglobins have an oxygen bound to them. As the concentration of oxygen starts to increase, like we're starting to leave the tissues, down here at the tissues, okay, the concentration of the tissues, there's not much oxygen because, of course, the tissues are using the oxygen, right? Hemoglobin gives up the oxygen to those tissues that need it. Myoglobin is still holding on to them fairly tight. Okay? When we get back to the lungs, okay, here's the lungs out here, we can see that essentially hemoglobin is binding it. What's happening is in these two different concentrations of oxygen, hemoglobin is changing. Myoglobin can't do this. So I'm going to describe hemoglobin to you, and then I'll describe why it doesn't work for myoglobin. Hemoglobin has 4 subunits. Okay? Each subunit in hemoglobin can bind 1 oxygen molecule. That's an O2, just like you saw in the diagram. Changing of the structure of that 1 for the binding of the very first oxygen in hemoglobin causes the interaction of that subunit with another subunit that doesn't have oxygen to change. That other subunit changes slightly, and now it's a much better binder of oxygen than it was before. So once 1 oxygen has bound to an empty hemoglobin, the next subunit is much more ready to bind than the first one was. Binding of an oxygen to that second one causes the third one to be even more determined to bind, and the third one causes the fourth one. See, we see a sequential movement now of binding of oxygen as a consequence of changes in these proteins, one after the other. So the further we go, the higher the concentration of oxygen, the stronger hemoglobin wants to bind it. This is really good, because when we're in the lungs, we've got plenty of oxygen. We want hemoglobin to grab onto all the oxygen that it can. First one binds, second one binds, third one binds, fourth one binds. Bang, we've got all four of them bound. When hemoglobin leaves the lungs and it starts going back out to the tissues, guess what happens? Exactly the reverse. Loss of one oxygen favors loss of a second one, favors loss of a third one, favors loss of a fourth one. This phenomenon that I'm describing to you in the binding of oxygen is called cooperativity. It's called positive cooperativity because it's favoring binding. In the reverse direction, it's called negative cooperativity because it's favoring the release. Now that's really cool. Okay? That is really cool. We're seeing the release of oxygen happening where it's needed. When it gets out here to the tissues and the concentration is low, hemoglobin is more than happy to give up its oxygen. Now, you'll notice in the tissues it doesn't go all the way down. So here's tissue concentration, and here's about how much it gives. There's about 35, 38% of the hemoglobins still have some oxygen bound to them. That sort of makes sense. We don't want hemoglobin to just give up all of its oxygen the first time it needs it, because the rest of the body is going to need it, right? If it did that, then as soon as it left the lungs, it would go to the very first thing it needs, and it would just dump its oxygen. So this allows hemoglobin to have some carrying capacity to where it's going to be needed later. Okay, that carrying capacity turns out to be really, really useful and interesting. Does everybody understand this graph? Yeah.
Could you explain the blue line, the hypothetical line?
So if you had no cooperativity, you would simply see binding solely as a function of oxygen concentration. That is, in this case, we're seeing the affinity of hemoglobin changing. Down here we've got low affinity. Up here we've got high affinity. So we see the affinity of hemoglobin changing for oxygen. If we simply had it binding only as a function of concentration, it would just go up as oxygen went up and then level off. Okay? So whenever we see this sort of, and it's exaggerated in other figures, but this sort of S shape that you see here, this S shape is indicative of the phenomenon of cooperativity. Cooperativity, binding of the first one favors the binding of the second one, favors the binding of the third one, favors the binding of the fourth one. We see cooperativity happening in a variety of ways. Okay, makes sense. Question?
And this happens, so you have myoglobin and hemoglobin, and it,
Okay.
you said that cooperativity level or cooperativity depends on like its binding to both.
Okay, so she just asked about myoglobin. So let me just clarify here. So hemoglobin binds. We see cooperativity because the first unit talks to the second unit, talks to the third unit, talks to the fourth unit, right? Those changes are communicated throughout the molecule. That requires communication. Myoglobin has one subunit. There's nobody to talk to. It doesn't have this phenomenon. Myoglobin simply binds, and that's it. So the myoglobin binding is sort of an exaggeration of what's here. It just binds very strongly at very low oxygen concentrations. That's good. That's what you want a battery to do, right? Myoglobin acts like an oxygen battery. Okay, so if you have that battery, it's there holding on to oxygen. And that's what it does here. Okay, I'm running out of time, so let me come back to that. I'll say a lot more about hemoglobin next time, and see you guys later. Okay.
Question?
Yes, ma'am.
You know when as you were talking about like the different like gel electrophoresis, like those types of methods, like today you just came back to myoglobin and stuff and hemoglobin, which is in the tertiary structure. In what I guess like structure or what structure they're in when they go when people put them and run them into
So,
like you said that you know how proteins have different structures.
Yeah. I'll take it if you just give me it away.
And then, what I guess like my main question is what structure do the proteins have to be in for it to be like run through electrophoresis?
Oh, well, generally for electrophoresis, proteins are almost always dissolved with SDS.
Oh,
So we gotta have that rod.
it just like denatures it and
Yes.
just turns it back to
Almost always, yeah.
Okay.
There are some techniques for proteins that don't do that, but they're more complicated.
Okay. And then I also had a question about methionine. Just like I'm curious, why is it always that methionine is always like the start?
Because it is. That's the way we evolved. And having a consistent start that's recognized across all of biology made great sense because everything has evolved from the earlier ones that were there. So the first ones had it, and everybody else has been doomed since then. So, okay.
Okay.
Let's go back and get out of the way here. Did you have questions?
Yeah, I have one quick question.
Yeah.
I'll just wait.
Okay.
It's kind of awkward otherwise.
Okay, sure. Yeah.
I think it was the guy next to me. Anyway.
Okay. Well, that would help if you tell me that. But yeah, appreciate it. Thank you.
Hey, Kevin,
Yeah.
a little bit about cooperativity. Did you say hemoglobin is releasing, but hemoglobin in the opposite type of way that it binds it? So when it releases one, it'll be more likely to release the next one.
So when hemoglobin binds one, it's when it releases one or binds one.
When it releases one, when it does the,
So
when it goes
the
up.
backward